Thanks for coming so early this morning to our workshop on stable isotope labeling techniques. And one of the key motivations we have for doing this workshop is that we feel like some people are quite scared of doing labeling studies. They see quite complex papers coming out on metabolite flux and things like that. And we're trying to get across the message that you can actually do stable isotope labeling experiments quite easily if you get your experimental procedures right from the start. So today what we're going to talk about are some of the basics of stable isotope labeling. Then we'll talk about a couple of case studies on how to do in vitro stable isotope labeling experiments. Then we'll talk a bit about some tools that we can use for stable isotope labeling data analysis. Then we'll talk about in vivo considerations, so doing carbon-13 labeling in the mouse or other animals. And finally, we'll end up with mass spec imaging and some closing remarks. So what we're not going to talk about today is four of these things. We're not going to talk about flux balance analysis. We're not going to talk about flux ratio analysis or kinetic flux profiling or MFA, metabolic flux analysis. That's quite complex approaches, and we're going to try and get across that you don't need to do that. Instead, today we're going to talk about qualitative fluxomics, or just simply put, stable isotope tracing experiments. Okay, so you all know what metabolomics is, I hope, but I'm just going to show this slide to talk a little bit about stable isotope labeling and in the context of one of the case studies that I'll talk about later. So here, my background is in tropical medicine, so I'll be talking in a little bit about the malaria parasite, Plasmodium falciparum. And Plasmodium falciparum lives in a red blood cell here, and what we've got here is we have the Plasmodium falciparum within a parasitophorus vacuole, which lives inside the red blood cell. And when we're doing metabolomics on Plasmodium falciparum, we're looking at the metabolites in the parasite, those that are in the red blood cell, those that are parasitized and taken up by the parasite. We're looking at some unanticipated metabolites, but we're also looking at those that are secreted out into the medium and other cells. But what we're talking about today is this powerful technique of carbon-13 glucose labeling. It doesn't have to be carbon-13. You can use other stable isotopes as well. And as I said, I'll be talking about how we do that in the Plasmodium falciparum parasite. For the first part of the talk, all of the stuff I'll be talking about will be on GCMS, not LCMS. We will be talking about LCMS approaches a bit later. And I've just got this one slide on GCMS because I know that LCMS really is the predominant tool for doing metabolomics. But just one small slide then. This is what a GCMS looks like. And on the right-hand side, sorry, I don't have a laser pointer, but on the right-hand side of the GCMS instrument, in that blue box, we have the GC oven. And what we do is we inject our metabolites into this oven, where it's in the top left here, we've got the, I'll just move over, hopefully you can still hear me. In the top left, just there, we have the inlet, and that's, oh, thank you. Which button? Yeah, so this is the inlet. This is sitting at 270 degrees, so all of our metabolites will, that are in liquid form will instantly volatilize into the gas phase, and helium will carry that onto this 40-meter long column here that's only got a 0.25 millimeter core running through it, and coating that tube is a 0.25 micron hydrophobic sticky gum to which our molecules will attach. The oven itself sits at 70 degrees, so everything that was in the gas phase at 270 will be stuck onto this sticky gum, and then all we do is raise the temperature of the oven, so small polar molecules will come off earlier, and then larger, less polar ones will come off later, and they'll travel through this 40-meter long tube, and then through here into the mass spec itself, okay? And what our results will look like is a classic chromatogram, as you'll be used to with LCMS, but we tend to have quite good resolution with GCMS. And actually, any one of these peaks is only 1 to three metabolites, unlike what you would see in LCMS. And I can tell that this is myo-inositol here, and I can tell that because of the characteristic ionization pattern here, this ion pattern, and it will always look like this. Because GCMS has got a hard ionization technique, electron impact ionization, this means that we will always see fragments. We very rarely see the molecular ion, but what is really good is that we'll see a ratio of fragment ions, and they will always be in this ratio. And so this is quite diagnostic. So this is the fragmentation pattern for myo-inositol, and you can see here we've got 318 here, which has got 4 carbons here, and this 305 ion has got 3 carbons. You'll also notice this TMS that I've written here. That stands for trimethylsilyl. We don't really need to know about that today, but that's just the derivatization agent that we use when doing GCMS studies. So this is what unlabeled myo-inositol looks like. If it was labeled, these carbons would become carbon 13 rather than carbon 12. And what we'll see is that we'll see some of that 318 is now 322, because that's 4 carbons that have become carbon 13, so the molecular weight goes up by 4. 305 goes to 308, 217 to 220, and so on and so forth. And what we could say here, just by looking at this, is, well, that's probably about 40% of all of the molecules are 322 rather than 318, so that's 40% label incorporation. So that's pretty much, in its simplest form, what we're doing here. But there's a lot of information that we can get beyond just this, so let me try to explain that now. So in this cartoon, what I've got is citrate, and you can imagine here that we've got the unlabeled citrate version takes up about 25% of all of the citrate molecules that we're measuring. Here, at +6, because citrate has got 6 carbons, this is about 75% of all of the total citrate molecules. And it kind of would look like this. So in this situation, you can say that 75% of the molecules are labeled. But in this case, still with citrate, again, 25% of the molecules are completely unlabeled. And here, we have 75% of the molecules are labeled, but only in 2 carbons. And that would look something like this. But again, you can say that 75% of the molecules are labeled. And then here, another example, 25% unlabeled, and then the rest is spread out among a number of different isotopomers. And here, again, 75% are labeled. This is something that you need to consider when you're reading metabolomics papers that have isotope labeling in them, because quite often people will say, oh, it's 75% labeled, or they'll show a heat map, and I'll be showing heat maps later on. But there's a lot of information that is hidden if you don't show or take into account these isotopolog patterns. Another way that we can talk about this is by looking at the number of carbons that are labeled. And in this top cartoon, if we look at the carbon pool, we can see that 75% of all of the carbons are carbon 13. And in this, only 25% of all of the carbons are labeled. And finally here, 50% of all of those carbons are labeled. So again, knowing exactly how to portray your stable isotope labeling results is really important, just as important as how you interpret the papers that you read and the talks that you will listen to later in the week. Okay, and that's the background over. So I'm going to go into a case study where we've looked at the malaria parasite that I introduced earlier. And this is quite an old study now, about 10 years old, but it does illustrate quite nicely what you can do with 1 or 2 stable isotope labeling experiments. Okay, so I won't dwell on this too much because I think most people have heard of malaria. It's still a prevalent disease all over the world. There have been vaccines that have been rolled out a couple of years ago, but we're always looking to develop new drugs because we'll always encounter resistance, so we always need to keep working on it. I'm just going to dwell a little bit on the life cycle because I'm going to be talking about some of these stages. And I mentioned earlier about the red blood cells, the erythrocytes, and how they are infecting our red blood cells. That's where the disease is manifest. So when you have the malaria disease, it's because of the parasite getting into these asexual stages here. So how does that happen? When you get bitten by a mosquito, it injects this sporozoite form of the parasite that infects your liver cells. And then at some point, after some rapid proliferation, they'll burst out, enter the bloodstream, and most of these parasites will invade your red blood cell and enter asexual reproduction, or something that we call schizogeny, okay? So we'll be just going around and around, growing and growing in the red blood cell. They'll burst open and reinvade more red blood cells. This is really quick, about 48 hours, and so you can imagine how a couple of rounds, you're going to soon have a lot of red blood cells that are infected. However, a small proportion of these, somewhere between 0.5% to 3% of all of these schizonts, when they reinvade a red blood cell, they'll enter a sexual stage. And we don't really understand the triggers fully for what happens here, but they'll enter this gametocyte stage. This is important, not for the disease that we encounter in ourselves, but it's important for transmission. So it's these stages that, when they're taken up, will enter the midgut of the mosquito and go through sexual reproduction to finalize the life cycle. And I'll come back to some of those stages later. This is the Plasmodium falciparum parasite itself. This isn't my figure, but it is still quite beautiful, I think. So I just wanted to show you here that we've got organelles just like the mitochondrion, that we'll all know about, being the energy powerhouse of most cells. And we've also got this organelle here called the plastid, or the apicoplast, which is kind of like a chloroplast that doesn't do photosynthesis. Instead, it does fatty acid biosynthesis and some other stuff like isoprenoid biosynthesis. Why am I talking about that? Well, that's important for doing our energy experiments and our labeling experiments, because in most cases, when we're doing energy metabolism, pyruvate is converted to acetyl-CoA via pyruvate dehydrogenase, which sits in the mitochondrion. This is absolutely essential for the canonical operation of a TCA cycle. You must have this. However, in the Plasmodium parasite, that pyruvate dehydrogenase is not in the mitochondrion. It's in the apicoplast, where it contributes to fatty acid biosynthesis. For this reason, for many decades, people thought that there was no TCA cycle in Plasmodium falciparum, but actually, all of the other enzymes are there for the TCA, so why are they there if it's not doing a TCA cycle? So we wanted to investigate that, and we've done that by doing stable isotope labeling. And here is a heat map, and I've already warned you about reading too much into heat maps, but here what we've done is a simple glucose labeling experiment and a glutamine labeling experiment, where we've added the glucose, and you can see these yellow boxes here. That's quite a lot of label incorporation, about 60%, getting all the way down to PET, lactate, pyruvate at the bottom of glycolysis. But when we look at the metabolites in the TCA cycle, you can see that there's not really a lot going on there, maybe about 20% labeling, but there's definitely labeling there. So already we were thinking, well, hang on, how is that glucose labeling getting in? It must be a TCA cycle. In the glutamine is a kind of reciprocal experiment where the glutamine comes in this way, and you can see that the glutamine is being incorporated into malate and aspartate. By the way, aspartate we use as a proxy for oxaloacetate. But yeah, so if we're looking at the malate, you can see that there's quite a lot of label incorporation there. And again, looking at alpha-ketoglutarate, which sits here, and succinate. So we're getting quite a lot of label here. So this is indicative that there's stuff going on in the bottom half of the TCA cycle, but actually not a lot in citrate. And again, there is stuff there, but it is saying that there's probably a TCA cycle. So we can look at that in a bit more detail, going back to our isotope log patterns, and we can do this by this. So when we've added carbon-13 glucose, it's got 6 carbon-13s. By the time that gets down to pyruvate, that's only got 3 carbons, so we're looking at 3 carbon-13s in the pyruvate. Acetyl-CoA has only got 2 carbons, so we'll be looking at a +2 for acetyl-CoA. And so what we see then With citrate, we would look for a +2 as every unit of acetyl-CoA is incorporated, and that will carry through the TCA cycle, where it will pick up another 2, so you'd expect to see this citrate becoming +4, and if it goes around again, it will become +6. So that's one thing we're looking for, a +2, +2, +2 pattern. We would also look for evidence of this. So PEP, or sometimes pyruvate, can be carboxylated to form oxaloacetate in the cytosol. And if that happens, this +3 PEP will become a +3 oxaloacetate, because it will pick up a cold or a carbon-12 atom from bicarbonate in the cytosol. Okay? That oxaloacetate can be converted to malate, which will then cross the mitochondrial membrane and can be incorporated into the TCA cycle if it's happening. And if this happens, what you can imagine here is this +3 malate will incorporate this +2 acetyl-CoA to make a +5 citrate. Okay, so there's quite a lot of stuff going on there. We're looking for a +2, +2, +2 pattern or a +5. And these are the actual results here, and you can see this is what we're seeing. So with the citrate, you can see +2, +2, +2, and then we're also seeing evidence of this +5 as well. In the succinate, of course, succinate's only got 4 carbons, so we're predominantly seeing the +2 and a little bit of +4. Similarly here and here, you can also see there's quite a lot of +3, which is again evidence of this PEP being converted to oxaloacetate. We can, of course, do the reciprocal experiment, as I mentioned earlier, with glutamine. And in this case, this glutamine will be converted to glutamate. That'll enter the mitochondrion. And here we're looking for fully labeled succinate, fumarate, malate, and oxaloacetate. And in this, we're not doing carbon-13 glucose labeling. So this acetyl-CoA will now be cold. So the citrate should be +4. And if it goes around again, it might become +2. And again, this is what we're seeing here. We're seeing predominantly +4 and a little bit of +2, which is showing that it is going around. Incidentally, sometimes there are situations where the TCA cycle could work in reverse. And if that was going to happen, this glutamate would be converted to alpha-ketoglutarate, which would go straight up to citrate, so we would see +5 citrate. And we don't really see any evidence of a +5 there. So we knew instantly that there is a TCA activity, and it's going around in a canonical direction. We wanted to test that by blocking, using this chemical inhibitor, sodium fluoroacetate. So if it was going in the canonical direction, we should expect to see an increase in citrate, and that's exactly what we see. If we just look at the blue bar here, which is that asexual stage that I was mentioning earlier, you can see that there's a huge increase in citrate in these cells. Interestingly, we were also looking at the sexual stages at the same time, and in this case, we get even more citrate, which was a first indication that there might be even more going on with the TCA cycle in the sexual stages. But does this matter? So we wanted to see, does it affect cell fitness? And what we saw, unfortunately, is no, it doesn't. So here, we've got our asexual stages, and just to walk you through this slide here, this is parasitemia. So when we're growing the malaria parasite in the lab, we're growing them at about 12% infection of red blood cells. We can't go too much higher than that because they'll crash out. So for you to try and understand this, probably the best way to think about it is that 12% is maximum infection, okay? And what you can see is that when we've added this inhibitor, it's gone down a little bit, but it's not significantly gone down. So this was a bit disappointing. So we looked again at those sexual stages. This is the result I showed you earlier. And now when we do the same experiment in the sexual stages, the gametocyte stages, we can see 2 things. First of all, there's been a complete switch in the preference of what carbon source is used. So you can see now there's a lot of label incorporation into these TCA metabolites using glucose, and not so much with the glutamine. So this was really interesting. But perhaps more significantly, what I didn't tell you earlier was that with the asexual stages, that was a 48-hour labeling experiment, while with the gametocyte stages, that was 4 hours. So not only is there a complete carbon source switch, but also it's happening a lot quicker. This is the isotopolog experiment again, so exactly the same as the one I showed you earlier, but this time I'm showing it as a time course, so it's a little bit more complicated. But let's just have a look at the succinate maybe, that's the easiest one to look at. So remember we're looking for a +2+2 pattern, and this is what we're seeing here. So if you look at the purple line here, that's +2, you can see that rapidly, after only about 20 minutes, we're seeing a lot of +2. And then that falls down a bit as it's overtaken by the +4. That's exactly what you would expect to see, is that +2 then becomes a +4. And we're also seeing, looking at the pink line, a lot of +3 as well, which is interesting. So again, we're getting a lot of that PEP carboxylation going on here. Now we can look at the citrate. It's the same pattern again, but a bit more complicated. The +2 immediately goes up and then comes down. Then we see the +4 coming through. And then after that, we see the +6. Again, exactly as we would expect if there's an active TCA cycle. And then we can also see here the orange line, the +5 as well. So this was all indicative that there is a very active canonical TCA cycle. But does it matter to the sexual stages? So here, it's the same experiment that I showed you earlier. So we're looking at the parasitemia. This is what happens when we're looking at sexual stages. We expect to see a drop-off, but also we expect to see a transition from red to blue to green. And that's the different levels of maturity of the gametocyte stage. This is important because it's this stage of the gametocyte that's taken up by the mosquito. So we're looking to see this for successful transmission of the parasite. And when we block using sodium fluoroacetate again, you can see that the parasitemia has really dropped off significantly, and those that make it to day 7 aren't mature anymore. So this told us that this was really having a significant effect. Okay. I told you earlier that we need pyruvate dehydrogenase, and yet there isn't pyruvate dehydrogenase in the mitochondrion of the Plasmodium falciparum parasite. So we wanted to know what was doing that activity. Excuse me. And I won't tell you all the reasons why we decided on this particular enzyme, but in the end we deduced that it was branched-chain ketoacid dehydrogenase. It has been seen to have a pyruvate dehydrogenase activity in some other organisms, so we thought that that might be what's causing the PDH activity here. And we showed this by switching to a different parasite, another Plasmodium parasite that causes malaria, P. burgii, and here we knocked out the BCKDH, and you could see that when we did this, our significant labeling that was going into the TCA cycle was ablated once we knocked it out, which again was indicative of that TCA activity. And this was the final piece of the jigsaw. So acetyl-CoA we can't observe via GCMS, so we switched to LCMS here. And here in the wild type, you can see this +2 acetyl-CoA that I was mentioning earlier. Acetyl-CoA has 2 carbons, so we'd expect to see a +2. And when we knock out BCKDH, it's gone. So this really showed that BCKDH was acting as a PDH. Okay. So I'm going to go back to the life cycle very quickly before I move on to some lipid work. And here are these different stages of maturity of the sexual stages in the midgut of the mosquito. So we're looking at the micro and macro gametes, the ookinete, and the oocyst. And when we were following those in the knockout, what we could see, just concentrating on the dark blue and the red here, we could see that in the knockout of our BCKDH, the amount of micro and macro gametes were significantly reduced. We could see that exflagellation and ookinete conversion was also reduced. But when we're looking at the oocysts, we could see that really that's very significantly reduced, which suggests that the TCA cycle really is halting transmission. And in fact, a lot of follow-up work over the next decade has shown that by looking at all of these different TCA and related enzymes, by knocking these out, you can really stop malaria transmission. Okay, I'm going to leave it there and move on to another related parasite called Toxoplasma gondii, which is, again, like Plasmodium, it's got that apicoplast organelle, and it shares many characteristics of Plasmodium falciparum, but it has a very different pattern. In fact, this is the world's most successful parasite. around 75–80% of the people in this room will have a Toxoplasma gondii infection. And just to quickly go through its life cycle, the obligatory host is the cat, so it must have a feline host at some point to produce oocysts. And these can easily be passed into an intermediate host that will include us, but mainly livestock is what we're worried about here. And the way that this happens is these oocysts are passed through the fecal material of the cat, which are ingested by livestock as they're grazing in the field. So cows, sheep, etc. can all be subject to Toxoplasma gondii infection. And this will manifest by these tachyzoite stages of T. gondii, rapidly dividing, causing tissue destruction, spreading, and then entering usually a dormant stage, perhaps in the muscle, perhaps in the central nervous system, where they'll just lay silent for many, many years. It's successful because although I've said tissue destruction and spreading, it doesn't do this at a great scale. However, in the immunocompromised and also pregnant women, it is a concern. And you may have heard that pregnant women are advised to stay away from cat litter trays, and that's exactly because of this, because you can easily get the infection through handling cat fecal material. Now, interestingly, just to sort of allay your fears, if you already have a Toxoplasma gondii infection, and as I've said, 75–80% of the people in this room will have that infection already, your developing fetus won't be affected by the infection. However, if you are Toxoplasma gondii free and you become pregnant and then you become infected with T. gondii, that can be a problem. It can lead to macular degeneration in the developing fetus and other more significant injuries as well. So it's something to be careful of. Okay, and just to look at what they look like in vitro when we're culturing them in the lab. So what we do is we use fibroblast cells coating the bottom of our tissue culture flask here. And these are the individual tachyzoite stages. And what they do is they invade the cell and they rapidly divide over a few days to 128 parasites per cell. And then they burst and they rapidly, they become very motile and swim along to the next cell that they can then invade, divide, burst open, and so on and so forth. So this is what we're looking at in vitro. I'll come back to this in a little bit. Again, this image is nothing to do with me, but it is a very nice indication of how we culture our cells. All right, so the TCA cycle was quite simple. This is a little bit more complicated now, fatty acid biosynthesis. As I mentioned earlier, this happens in the apicoplast. And I'm not going to dwell on this too much. You don't need to know all the details. But just to show, you've got glucose going to PEP, and in that PEP, it enters the apicoplast, where it enters this quite complicated fatty acid biosynthesis pathway. And what happens here is this is a fatty acid chain that grows, and I'll show you that in more detail in a sec. And at some point, it leaves the apicoplast, where it then can be elongated. And that happens on the surface of the endoplasmic reticulum by these elongase enzymes that I'll talk about a little later. Okay? So what we've done, and I'm not going to talk about all of these, is looked at mutants of all of these enzymes to try and work out what's going on with fatty acid biosynthesis in these parasites. So when we're doing a labeling experiment, again, just like I showed you earlier, we have the glucose coming in here, and it enters this way. A malonyl ACP here becomes a growing fatty acid chain. Again, it's units of 2. 2 carbon-13s are added. So just like the TCA cycle, when we're looking at fatty acid biosynthesis, we're looking at +2, +2, +2, +2. At some point, that fatty acid will leave the apicoplast, where it can then be elongated, and this ACCase2 enzyme converts acetyl-CoA to malonyl-CoA, and 2 of these carbons are attached to the growing fatty acid here. So again, we'll see +2, +2, +2, okay? The interesting thing that we can do, and I'll show you this result in a sec, is do carbon-13 acetate labeling. Now, this is a really powerful tool for us in the, when we're looking at the Toxoplasma gondii parasite, because acetate cannot enter the apicoplast. So while when we're doing a glucose labeling experiment, we label both the fatty acid and the elongation portions of fatty acid biosynthesis, when we're doing an acetate labeling experiment, we only look at the elongation. So that can be quite powerful. Okay, so I'm just going to spend a couple of minutes now talking about how we do a labeling experiment. And Shauni will talk a bit more about other considerations later on. But this is something that you really need to know the biology of your organism or whatever you're looking at when you're designing your experiment. So I mentioned earlier that these parasites take about 48 hours to divide and then burst out. That means when we're looking at fatty acid labeling, we've got a 48-hour window maximum to look at label incorporation. We can't do more than that. Why? Because once they burst out, they don't do any fatty acid biosynthesis anymore, because they've switched. All they need to do is become motile to invade another cell. So we've got 48 hours. However, at the same time, we're not doing knockouts of these enzymes. What we're doing is we're doing knockdowns. And we do that using a genetic mutant, which is under anhydrotetracycline control. So we add anhydrotetracycline, which will stop the production of the gene and the protein, or the enzymes that we're looking at, in this case, the acyl carrier protein. But that takes time as well. So generally what we do with the Toxoplasma gondii experiment when we're looking at lipids is we add the ATC for 24 hours, and then we start our carbon-13 labeling. It's important to mention that because when you look at the results I'm about to show you, you've got to remember that there will still be some of that enzyme activity lurking around. So it's not like we're going to see 100% labeling and 0. We're going to see a knockdown. Okay. So this is what we see. So just for those of you who aren't aware of fatty acid nomenclature, what we've got here is saturated fatty acids here. We've got the C14, so that's a 14 carbon fatty acid, and the 0 means no double bonds. And here we've got monounsaturated, so one double bond. So for all of the results I'll show, I'll organize it like that. So increasing chain length from left to right, the saturateds and the monounsaturateds. And what you can see here is that when we've added the anhydrotetracycline, so knocked down the acyl carrier protein, you can see that there's a real significant drop in the label incorporation here, and also we can see that here as well. Interestingly, we don't see a great deal of reduction in the 26-1, but this makes sense because remember, there is a lot of fatty acids already produced, and remember what I showed you earlier, that when we're doing carbon-13 glucose labeling, that can also go into elongation. Okay, so this is showing that there's still elongation going on here. Here's a pattern of the C14 fatty acid. So this is the unlabeled control, and here is the wild type, if you like, that's been labeled. And you can see this +2, +2, +2, +2, +2 pattern that I was telling you about earlier. And this is really the characteristic pattern that we'll look for in all of our studies. And then when we've added the ATC, so in the knockdown, we can see that it's almost completely ablated. Okay, so that was ACP. What I'm going to talk about now is ACCase2. So ACCase2, just to remind you, converts acetyl-CoA to malonyl-CoA, and this is what enables our elongation. So if we knock out ACCase2, we're not affecting fatty acid biosynthesis, but we are affecting the elongase reaction. Okay? So what we might expect to see is just labeling in the fatty acid that has come straight out of the apicoplast. So let's show you this then. So this is our labeling experiment. And again, in red, we've got the wild type. And in the sort of pale pink there is after we've knocked down the enzyme. And the first thing to look at here is in that carbon-14, you can see very little effect in label incorporation, which is exactly as you'd expect, because carbon-14 isn't elongated. Similarly with carbon 16 here, the 16-length fatty acid, again, not a significant drop, well, it actually is, but there's not a big drop in label incorporation there. Again, as we might expect if carbon 16, or the 16-carbon fatty acid, is what is being produced by fatty acid biosynthesis. After that, you can see there is a drop in the 18, in the 20, 22, so on and so forth. So this suggests, just from this, that it's likely that carbon 16, or 16 carbon length fatty acid, comes out of the apicoplast and is then elongated to the 18 carbon. I'll go through this in a little bit more detail shortly. Here's the relative abundances. All I wanted to point out here that really, even though all of the abundances have gone down, look at the monounsaturated ones here. They've really been affected by the reduction of elongation, as you might expect again, because if you can't elongate, you're not going to produce these molecules. So let's look at the labeling patterns. And you can see here carbon 16-1. So I'm just going to show the monounsaturated ones in the interest of time. And you can see this beautiful +2 +2 +2 pattern all over here, suggesting that these are really nicely labeled. C18-1, pretty similar. By the time we get to C21, you can see there's a big +2 here. And that is indicative of that first step of elongation. Similarly here with the 22, you're seeing a +2 +2, so that's 2 rounds of elongation, 3 rounds of elongation, and so on and so forth. But when we knock out the ACCAs, or rather knock it down, you can see that the C16-1 is not affected at all, but we're seeing a lot of labeling gone in all of the others, again suggesting that the ACCAs2 is stopping this elongation. I told you about the acetate. So let me just go back, sorry. So if we do the same experiment with acetate here, we'd expect to only see labeling in the elongated forms. So first thing to look at down the bottom here, the abundances, as you would expect, they're the same, because it is the same experiment. We've just knocked down the ACCAs2 and used a different stable isotope here. And also look at the label incorporation in these shorter fatty acids. There's not much at all, as you'd expect, because that's what's produced by fatty acid biosynthesis. It's these ones that are longer where we're seeing the elongation coming in. And in the knockdown, again, you can see there's a great reduction in the level of label incorporation. Just looking at the isotope log patterns again, you can see this very nice +2, +2 +2, 2, 2, 2, so on and so forth. So again, we're looking for that kind of pattern. And this is largely reduced in the knockdown. Okay, so my very final piece to just talk about is looking at even more depth. And here we've got 3 different elongase enzymes, A, B, and C. And using stable isotope labeling, we were able to tell exactly the role of each one of those. So here, what we've got is elongase C. And you can see that here, what we've got is a complete reduction in the labeling of 26-1. But it hasn't affected the label incorporation of any of the others. And you can see the abundance has gone down. So this was indicative that elongase C is responsible for that last addition of carbon units to the growing fatty acid chain. Elongase B, you can see there's a significant reduction here, here, and here. And that was telling us then that it's responsible for anything bigger than 18. So from 18 to 20, 20 to 22 is elongase B. And with A, it's a little bit of a more muddled story, but I think the take home here is that you can see there's lower label incorporation in quite a lot of the fatty acids. So that was suggesting that this was responsible for the first stages of elongation from 18 to 20. Okay, and this is my it's my penultimate slide, I think. I just wanted to highlight that fatty acid biosynthesis really is a potential drug target for Toxoplasma gondii and also the Plasmodium parasite, the malaria parasite. And these are just some of the drugs that target apicoplast metabolic pathways, including fatty acid biosynthesis here. And doing stable isotope labeling has just redoubled the importance of these particular pathways. How do we do this kind of data analysis? I'm going to hand over to Dave de Souza in a second, who will tell you about the way that they do LCMS stable isotope labeling data analysis. I don't have time to show you all of these details. I have brought this program with me, so if anybody's interested on how to do GCMS data analysis for stable isotope labeling, we've developed our own software, which we call Manic, and this here is just simply the citrate ion for all of the samples that we were particularly looking at, and we can at a glance see the level of label incorporation in this little summary here. You can see this is actually a time course, and you can see there's greater label incorporation in these later stages. And we can have an at a glance sight of the abundance as well. So this makes data analysis very straightforward. So I'll leave that up quickly if anybody's interested in looking at it, but also just come and see me afterwards if you're interested in that at all. And now I'm going to hand over to Dave to talk about something that sounds very similar to Manic, his LCMS data software. So Dave, over to you. That's the laser pointer. And then that's forward.
All right, thanks very much, James. So, yes, James has given you a fantastic grounding into why stable isotope labeling experiments are great and full of really useful information that really helps you interpret what's happening in the metabolism of your area of interest. So, of course, having convinced you that you should be doing these experiments, there's ways then to help you take your data from your instrumentation and get it through into a pipeline that will get out the information that you require to get all of those lovely figures that he produced earlier. So I'm talking about Maniac, which, yes, sounds a lot like Manic, which is an accessible LCMS stable isotope tracer metabolomics pipeline that we've developed at Metabolomics Australia in Melbourne. So, of course, James has already really given you a background into the motivation behind why we'd want a nice pipeline like this. Stable isotope labeling, as you can see, has low experimental overhead. You're only really adding in the labeling to what would be your traditional experiments in general, but you then get a really high information return. So really what we wanted then is a really easy to use standardized data analysis tool that really outputs all the sets of information with very little handling and error-prone kind of steps. So what we've put together is really just a variety of data processing tools with the key aspect of needing to correct for natural isotope abundance. So as James has already taken you through, stable isotope labeling, has an abundance of information. Of course, you can get from it the total metabolite pool, which is your traditional steady-state metabolome information that we're all familiar with. But then, of course, you want to go into the next levels of it, which is the percentage fractional contribution. So what is the percentage labeling of that pool? What does that mean in terms of the mass isotopological distribution pattern? Where has that carbon gone? And then you can use that to start determining pathway directions, as James really nicely demonstrated earlier, looking at things such as the directionality of that carbon through glycolysis and into the TCA cycle, whether it's gone in from via acetyl-CoA, so a +2 pattern, or directly into oxaloacetate, giving you a +3 type of pattern. So heaps of information that really starts to dissect what's going on. And of course, you want to then be able to provide this information back to the people that are interested in what's happening with your biology. So the first step of any data analysis, once you've pulled out your data from your analytical experiment, is to correct for natural isotopic abundance. So as we know, there is natural isotopic abundance in nature. So with carbon-12, you'll then have carbon-13 abundances that you will see. So on the mass spectrometer, something like what we're seeing up here for citrate, you'll see your natural carbon-12 mass, but then you'll also see small amounts of +1, +2, and even a little touch of +3 abundances on the mass spectrometer. So in terms of a labeling experiment, we want to be able to accurately portray what that incorporation is when the carbon-13 that you're feeding in shifts from the unlabeled into the labeled mass isotope logs. And so to do that, we then need to correct for these abundances and subtract them from those mass isotope logs. So having gone through and done that step, you will then be able to actually have a correction where you've corrected now to what is the actual carbon-12, or whether it's +1, +2, +3 carbon-13 incorporation. So traditionally, when we were doing this before putting together this pipeline, it was a huge multi-step process. We first had to take our input from our standard LCMS Metabolomics processing tool, so your picking and integration tool. Take your output from that, basically a data matrix, go to the next step of correcting for natural isotopic abundance, then go on and take that information and calculate the percentage fractional contribution, calculate the mass isotopologs, and then output it to a standard result data set. And so this is what it kind of looked like. These multiple, each sheet was an individual multiple step. And what that meant is that there was a hell of a lot of heavy lifting. That also resulted in likelihood of errors because there was so much manual manipulation of that data to try and get out all the information that you wanted. And so just to make it easier for the people in my team, we went through to put together the MANIAC pipeline. So what that stands for is Metabolomics Australia's Natural Isotopic Abundance Correction pipeline. So what we do is we use Leuven for our peak picking and data curation. So it's quite straightforward. This allows for any vendor-based data acquisition to be then converted using ProteoWizard, passed into Leuven for peak alignment, picking and curation. And what we're doing with that is to be uploading a targeted data, list of metabolites and their mass isotope logs that we expect. So in general, stable isotope labeling is a very much a targeted approach. So with that, we're then able to export these peak tables and metadata in a CSV format. And we then put this into IsoCorrector, which is an R package that corrects for natural isotope abundance. So what we've done to now make this all a much smoother single approach is to wrap it, it's an R Shauni wrapped package that then allows you to take the Leuven output and put it straight through into IsoCorrector, which then takes it through after the natural isotopic abundance correction to then tabulate and report all of the information that we'd want out of that. And we've also then added in just some very basic visualization so that you can start to have a bit of a first pass interpretation of your data. All right, so as I said, with that first step of the Leuven output, this is just a typical screenshot of what you'd see with Leuven. It's great because it allows you to visually validate every peak that you're integrating. So especially as you're looking to see those mass isotope logs as they build up, you can visually validate that you're picking the right one and that you're not picking something that's really noisy or the wrong peak completely. What that allows then is for a CSV output with your compound of interest, so in this case glutamine with as your carbon-12 M0 and the molecular formula for it. So with that molecular formula you can then predict what your remaining mass isotope logs are going to be. So for C5 you'll have up to 5 mass isotope logs. Added in there, you can also add in a group if you want to also further down the track be able to sort your information into cohorts as well. So that's a very simple, straightforward output, and that output then goes straight into our Maniac package. So as you can see, this is a very simple looking package, and that's all we wanted it to be. Essentially, you then choose that data file and hit run. What it does is it automatically detects what tracer type is in there. And this, because of isocorrector, you're able to do carbon-13, N15. We've added in deuteration. And if you're using an ultra-high-res instrument, you can do dual labeling with carbon-13 and N15 together. So once you hit run, it's a pretty straightforward, short amount of time. And then what it does is to spit out an output which will have a multitude of sheets with different information that you can then use. So you've got the corrected data, normalized metabolite pool, percent mass isotope log distribution, and so on, as you can see down there. And we then go on into some basic visualizations as well. So as part of the package, we have added into it any error handling if anything has not been set up correctly in the first instance. We've also added in the ability to normalize your pool size data if you want. So you can have any kind of normalization factor added in there, whether it's something like cell number. We've been going a lot with protein quantity. So we'll do a protein assay on our cell pellets after we've done our extractions. And you can add that in there, and that'll go on to normalizing that pool size data. So natural abundance correction, metabolite pool, you get that through summing up your mass isotope logs, and as I said, an addition of the normalization factor. We also add in the percentage fractional contribution. This can be done by cohort if you've added that in as a group, the mass isotope log distribution patterns, and again by cohort if you are interested in doing that. So in terms of very simplistic data visualization within the tool, this is essentially what we have. Some basic bar charts. Again, they're certainly not publication quality, but what they are is an ability just for you to be able to have a really quick look at your data and start to see if there's, patterns emerging to help you already start to get a sense of what's going on. So again, bar charts just to compare your pool size, so that's your typical steady-state metabolomics. We can move into the mass isotope log distribution patterns and start to look to see whether there is shifts in the labeling and how that compares between groups. And then the fractional contribution of that labeling as well. So all of this information then comes out, as I said, into this exported Excel format with multiple tabs with all of that information contained within it and well organized and ordered. Basically, all of this information is really what you require then to be able to go through into further packages or any of your basic tools such as Excel or Prism to continue to make sense of this data. So really, what we've done is to really just make a very portable solution. You can take these through the area file, can go straight into something like Metaboanalyst, and you can create your standard steady state outputs. You can then take your pool size and your fractional contribution and start using tools such as Prism to make use of both pool size and fractional labeling to give you something like these bubble plots that incorporate 2 bits of information. I've also got up here Travis Pies, which is from Bart's lab. The outputs from this tool are also designed to go straight into Travis Pies as well. And really, you know, you're not limited then to those types of tools. You can then just take all of that information and choose to represent it as you like. And this is just an example of a figure from a publication that looked at using the mass isotope log distribution patterns to give you a schematic of how that worked, as well as comparisons of the fractional contribution, and then also things such as looking at the mass isotope log distributions and comparing them on single metabolites, or taking them and going on to do ratios to be able to compare between conditions. So really, with those outputs, the sky's the limit for how you want to express and show that data. So just to summarize then, you know, we've already established that stable isotope labeling is a hugely powerful technique. It's going to give you insights into metabolic pathways and molecular incorporation that you're not going to get just from steady state experiments. Realistically, it is low overhead, experimental overhead, with a really high return. In terms of Maniac, it's web hosted and easy to use, or can be downloadable. You have these standardized results, and it provides you with these key outputs for downstream interpretation. My email address is there. Please feel free to contact me. You can also go and take a look at a poster being presented by Nadeem, who's from my team. That's poster 1079. So it's a tracer metabolomics based poster, and he's used this tool and others to generate that data, so you can always have a chat to him about how this works. So that's pretty much it for the LC pipeline. We know that there's a lot of people using LC, so please do come and have a chat. And Shauni is going to take over now and give you even more reason for why you should be doing these types of experiments. Cool.
Okay, so I think most of you will not know me. My name is Shauni. I'm a postdoc in the lab of Bart, but I'm also a consulting scientist in the core that he runs, which basically means that people see me at the very beginning of their experiments, mainly tracer, where I help them with the setup of their experiments, but also at the very end when it comes to the biological interpretation of this data. I will give you a quick guide on in vivo stable isotope tracing. If you're expecting a protocol after the next 10-15 minutes so you can start your experiments, it will not be the case. If anything, you will have even more questions, but I'm here to help you go through all of these. So we've had our introduction on stable isotope tracing, but let's take a step back because it may make you understand a bit better where in vivo stable isotope tracing becomes useful. So if we look at our different types of metabolomics, we can discriminate regular metabolomics, either untargeted or targeted, in vitro tracer metabolomics, and lastly, in vivo. And all of these have their different place in the landscape of metabolomics. if we start with regular metabolomics, I think the scope here is mainly broad and hypothesis generating. I think it's very useful for biomarker discovery, for very general phenotyping, but it doesn't give you any information on fluxes. It gives you a snapshot, so you have no idea on activity, like I said, and the interpretation is rather a correlation more than mechanistic insights, and the complexity is low. So most people, when they start with metabolomics, this is where it happens. Then we have in vitro tracer metabolomics, which is very targeted and mechanistic, and it will give you information on fluxes. But in vitro, we're working with a controlled and artificial environment. So, for example, in cell culture conditions, we're working at normal oxygen levels. Is this also the case for the cell type you're using in normal physiology, or the medium type that you use makes a very big difference. So that's why we have to be very careful. So it is mechanistic, but it's limited. And I would say complexity is medium. And then lastly, we have in vivo tracer metabolomics. So this is very targeted, but also physiological. We will have our flux insight, and it's the whole body a very dynamic context. And the mechanisms we get here, it's more realistic because we're looking in the animal or in the human itself. The complexity, however, is rather high, and so there's a lot of variables that we need to take into account. So being part of the core, I know that a lot of clients, when they get their data and it's not what they expect it to be, they love to blame us for it. They're like, something went wrong in the run, but actually, especially in vivo tracing, success starts long before the LCMS. Because good experimental design will mean you will get meaningful information. My PhD supervisor loved the quote, shit in is shit out, and I think it's exactly the same with experiments like these. So before you start, I want you to ask yourself 3 questions. Which tracer should I use? How should I administer it? And is there any other biology that needs to be controlled? So when we want to select our tracer, it's important to keep 2 things in mind. Which pathway do you want to look at, and which tissue are you interested in? So unlike regular metabolomics, untargeted, for example, you can just have an overview of everything. Here it's very important that you know upfront which pathway you want to look at. Is it glycolysis? Is it the TCA cycle? Is it the pentose phosphate pathway? And apart from that, you have to be sure that this pathway is active in the tissue that you're looking in. So for example, we know gluconeogenesis is very high in the liver, but if you're going to look at bone, for example, you will have very limited gluconeogenesis. And so there's some examples of tracers that I list here. So we have uniformly labeled glucose, which give you an overview of central carbon metabolism, glycolysis, TCA cycle, all of these. Then we have glutamine, glutaminolysis, again, TCA cycle or reductive carboxylation, acetate if you're interested in lipogenesis, and you can even use your positionally labeled tracers. So for example, 1,2 glucose if you want to discriminate between pentose phosphate pathway and glycolysis. If you say, okay, I don't really have one specific pathway that I'm interested in, I want to have a nice overview, then I think uniformly labeled glucose is the way to go, because it will really give you a very broad overview of central carbon metabolism, and based on that data, you can then design more specific follow-up experiments. So once you have chosen your tracer, you have to think about how are you going to administer it, because compared to in vitro, you just add it to the cell culture medium. When we're using, for example, mice, there's different ways where we can do this. So I've listed them here. You can give them orally, via IP injection or IV. And again, each one of these has their benefits and places in vivo tracer metabolomics. It's not that one is better than the other. So orally, I would say it's quite easy, depending on whether you give it via diet or gavage. The absorption speed is rather variable because, of course, it depends on digestion, and you do have your first pass metabolism from the liver. The physiological relevance is very high because, of course, we also get a majority of our nutrients through the diet, and the kinetics are slow and variable. So you have to really think about, okay, how long will I put the animals on these diets, and it will take some optimization as well. Then we have IP injection, which is very easy. The absorption speed is rather moderate, and you only have a partial first pass metabolism. Again, the physiological relevance, I would say, is moderate, and the kinetics as well. So it's this in-between method, I would say, IP injection. And then lastly, we have IV injections. These are technically more demanding. So you need specific expertise to do this. The absorption speed is very fast because you have immediate systemic availability. There is no first pass metabolism, so this is also a benefit. The physiological relevance is again high, and the kinetics are very fast and controlled. So we now have our tracer, we know how we want to administer it, and then the next question would be, okay, how much will you administer? And here it's again a bit of a balance, because of course we don't want to give too few tracer, because then we will have a poor signal to noise ratio, but also we don't want to go too high so that we're perturbing endogenous metabolism. If you think about glucose, normal physiology, we're around 5.5 millimolar. If you then would administer 20 millimolar of glucose, we're inducing hyperglycemia in your model, and of course you're going to get more of a diabetic response than anything else. So it's very important that you find this optimal dose where you have a good signal, but that there's no perturbation. And this will require a lot of pilot studies, and you always have to keep in mind that a tracer is a tracer and it's not a treatment, so it should not be inducing another biological response in your model. Then there's another question we can ask ourselves, which is, are we going to give just a bolus, or are we going to go for infusion? So if we go for a bolus or a single injection, this is very simple, quick, and cost-effective. However, it is for short-term kinetics because it will capture these early onset metabolic events. It's a spike, so it's more a perturbation compared to infusion. You also have transient labeling, so you will never really have steady state when you're giving a bolus injection, and that means that the timing is critical. So I want to show this with an experiment that was performed, where mice were injected with uniformly labeled glucose, and we waited for 4 hours, and we looked at plasma and the kidney, and then we wanted to see, okay, how does the labeling look? And after 4 hours, we actually didn't have any labeling anymore. So it was like, okay, this is not good, what we think happened. So then when they repeated the experiment, they went for a bit shorter time points, which 5, 10, and 30 minutes. And you can actually nicely see that already at 5 minutes observed incorporation in lactate in the kidney. But that it's also decreasing quite fast, showcasing that your timing with these bolus injections is very critical, because you might just lose the window for where you have the nice data. And that's then where infusion comes in, where we give a constant stream of tracer. This will require catheterization and infusion pumps, which means, again, more specific setups that not everybody has available. It's useful for long-term kinetics, and because because we have the animal on this infusion for a longer period of time, it would also minimize the metabolic perturbation, and you will be able to reach a steady state, meaning that also flux analysis is possible. But like I said, it's technically demanding and it's also very time consuming. And here's, I think, a nice visualization of comparing bolus with infusion, because with a bolus we will start very high, but our label will decrease rapidly over time, and you have to find the time frame where you have still a nice label. But for infusion, you have to wait a bit before you're reaching this plateau, and that's where you want to do your analysis. And then I labeled this slide everything else that can mess up your experiment, because these are more biological parameters that, especially coming from biology background, some people often forget that this could influence your tracer as well. So the first one is actually the feeding status. I will talk about mice, but this of course also counts for if you want to do in vivo tracer studies in animals. But there you have to make the choice whether you want to do this in a fed or fasted state. So I know a lot of experiments in mice are happening in fasted states because of certain reasons, but your hunger hormones can have very big influence on your tracer outcome. So sometimes it's better to just opt for a fed state. Then again, if you are performing other animal experiments in parallel, you choose the setup that is the most relevant for your model. So if you have obtained all your data in fasted states, then of course for the tracing you would also go for fasted state, but it's just something to keep in mind. Also, the circadian rhythm is something to take into account. And that's especially when you're doing in vivo tracer in multiple animals, that you have very consistent timing, and you also go for fast tissue collection. With consistent timing, I mean if you're, for example, performing infusion experiments and you say, okay, I want to infuse my animals for 2 hours and I can only do 2 at a time, but I have very big batch, that you plan these experiments on different days, and that you don't say, okay, I start at 8 in the morning and I just keep infusing different animals throughout the day, because this will be another variable added that can complicate your data analysis. With fast tissue collection, it's important that once you stop the tracing, you collect the blood and plasma as fast as possible and snap freeze any organ that you might be interested in, because it's not because the animal died that your tissue all of a sudden becomes dormant, so things will keep happening. Then also the mice, it might be a very logical one, but take into account age of mice and try to keep this as consistent as possible. Also sex of the mice, because there will be differences in male to female because of the sex hormones, and also the strain. So if you're using black 6 mice versus bulb C, certain tracer profiles will also look different. Then a variable which some people might underestimate, it's actually us, because we will induce stress, whether it's the handling of the mice, restraint, and the anesthesia. I've known people that perform in vivo tracing where they don't anesthetize the mice, and they go for an IV injection. The animal will have a lot of stress during the handling. And if for one animal you are immediately finding the tail vein for the injection, but for another animal it takes 10 minutes, there will be a difference in their tracer profiles because of these stress hormones. And then lastly, the environment, meaning temperature, housing, and the physical activity of your animals. If you're performing your experiments over multiple days, in most animal facilities, temperature is very consistent, but try to keep this in mind as well when doing your experiments. Then something else that I want to give along is that because you're working in a full body model, your organs, they don't function on their own. You have constant communication between those organs, which means that your tracers are recycled and exchanged between organs. So lactate generated in one tissue might be a source of carbon for another tissue. And so the labeling patterns that you see, it reflects systemic metabolism and not just local activity of this one tissue. And this, of course, makes everything a bit more complicated because you will have dilution, scrambling, and misinterpretation. So you just have to keep in mind that due to this inter-organ metabolite exchange and redistribution, that your labeling patterns will be a bit more complex and might not look the same as when you're doing an in vitro tracer experiment. So before you blame the mass spec for having shitty data after in vivo tracing, make sure you define your biological question, that you select your tracer, choose your administration route, define all your other biological parameters, do a pilot experiment, and I've put pilot experiment, but I actually mean experiments because there will be a lot and expect to optimize a lot. It will take multiple rounds of optimization, and I know sometimes we want to go as fast as possible, but especially for these experiments, it will pay off if you take a bit of a slower pace. And then I want to end with what if we didn't need in vivo stable isotope tracing, which is actually some of my own work. So if you're still here on Wednesday, I'm giving a presentation on high throughput ex vivo stable isotope tracing using tissue homogenates to model in vivo metabolism. I will not go into detail there, but if you're interested in how you could avoid in vivo stable isotope tracing, then make sure to pass by. So if you would ever have any questions, also feel free to contact me. I'll be happy to help you. I cannot promise I will solve all the problems, but I'll try my best. And now I will give the floor to Bart.
Okay, good morning. I'm going to continue with the slide of Shauni. Where we have done actually an in vivo experiment. We infused tracers into the mice, could be any tracer. What we will not do now is homogenize the tissue. We're not going to take the kidney out and see what the tracer profiles are in the kidney. Oops, sorry. Yeah. I thought about it and I was like, yeah. So what we will do here is we're going to keep the tissue intact. So the mice or the rats have been fed, infused with tracers. We took the specific key organs out, and we're going to slice them. We're going to section them. We're going to analyze these sections. So this is the field of imaging mass spectrometry. There's two main technologies. I'm not going to go too much into details. MALDI, matrix-assisted laser desorption ionization. You shoot with a laser at the tissue. And then you have DESI, desorption electrospray ionization. You're going to spray a solvent on the tissue. The main difference between these two is this is really soft ionization. You're not going to damage the tissue in any way. So it is very compatible with all kinds of technologies, H&E staining, spatial transcriptomics, blah, 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 and so on and so on. Here, they say it's semi-destructive. We have the instrument in my facility. It is destructive. You see the laser marks on the tissue. When you then go for H&E staining or spatial technologies, you do see the burn marks. So it's really, yeah, choosing which one you would like to have. In addition, laser-based systems are expensive. When the laser breaks down, you need to replace it. This is quite a big cost. With the DESI, that's not the case. Okay, so how does So my group has a DAISY-based system from otters. It's a DAISY MRT. How does it work? We have a spray needle here. It's going to spray a solvent on top of the tissue, and then you get secondary droplets. In essence, the first droplets take metabolites along, and they bring them into the mass spec. There's a whole theory behind this. Not going to go here in details, but with playing with different solvents gets you also different metabolites. So if we look at the two technologies, it starts all here. We took our organ, we sectioned it, and we put a section onto a glass slide. If you want to do MALDI, it needs to be a conductive slide, quite expensive. If you want to do DAISY, it's a normal glass slide. So the tissue for MALDI is then brought into a sprayer, and we're going to apply a matrix onto it. So here we have to choose very carefully. Each matrix that you can use in MALDI has a specificity for certain metabolites. So again, as Shauni pointed it out, with these systems you really need to think upfront, what do I want to study, which tracer, which pathway, what are the key targets. And then of course after the matrix has been applied, you can bring it into the machine and measure. With DAISY, it's pretty straightforward. Once you have the tissue section, you bring it into the machine, and that's it. So my group is focusing heavily on ischemia reperfusion damage in the kidney. Why the kidney? Animals have two kidneys. This is like a brilliant system where you have the ideal control, the left kidney or the right kidney, and then the other one gets clamped for an amount of time. The clamp is removed, blood flows again into the kidney and creates this damage caused by oxygen and radical species. So two images here. This is focusing on, I think if it's not mistaken, it's lactic acid. You can see in the damaged kidney a different zone that creates this lactic acid, and that's just the lactic acid abundances. We also took this section and then did H&E staining, so we can overlay, study which damaged area is present, and does that correlate with the metabolic signatures that we see with the DAISY. For now, we see over 80 metabolites covering amino acids, nucleotides, fatty acids, organic acids, so central carbon metabolism. Again, this is a zoomed-in image of lactic acid. You can clearly see it in the medulla of the kidney, not in the cortex. And this is what the technology does. It shows you where are certain molecules, small molecules, located in a tissue in specific conditions. Of course, this is not with tracers. And there comes a very interesting thing. When we combine imaging mass spectrometry with tracers, it really brings us as close as possible in vivo to look at metabolic events. So this combines everything, bringing in the isotopes, let's say the +2 of citric acid. It tells me a biochemical story. And if I see the +2 of citric acid in a specific area of my organ, it does tell me that there a specific biochemical activity is present, and then it's up to us to dissect what does this +2 mean, and so on and so on. Initially, we thought we're going to do bolus injections. Shauni also said this is challenging, and we used D7 glucose just to see when do we see the peak of the labeled molecule appearing. I just want to point out this. Already after 3 minutes, we only see 9% of this tracer. So 3 minutes on an animal experiment, that is really, really challenging. So we decided to switch into infusing the tracer into the animal so that we have sufficient amounts of tracer present. Here as well, we had collaborators that have done these IP, so intraperitoneal injections, with a bolus. It was very challenging because they did, I think, about 50 animals. The variation between each animal is massive. And on top of that, if the tracer profile is very low in the organs of interest, it also becomes problematic to understand what's happening. If you only have 9% of labeled glucose in an organ, you can imagine that if we go through glycolysis and the TCA, we quickly will lose the tracer. So it's not going to give us that much information. This is really what tracer imaging is about. Looking at all the isotopes, where are they located in a spatial way in healthy and diseased systems. So here, I want to put your attention here to glutamic acid. So we use 13C glucose. This is a kidney. This is the unlabeled glutamic acid, and you see a signature of the +2 glutamic acid. This tells me that here glucose went via glycolysis into the Krebs cycle to alpha-ketoglutaric acid, got converted into glutamate. This area is producing glutamate from glucose. So imagine now this is just a couple of these metabolites with isotope profiles. We can do that for hundreds of them. So we really show biochemical activities happening in the organ in specific anatomical areas. For us, with the kidneys, we're mainly interested if this is a damaged area, what metabolic pathways are at play, and can we then see if we can stop it or intervene with them. So this is the beauty of in vivo tracer imaging mass spectrometry, showing the biochemistry on site in which anatomical area. That was the kidney. We can do it on brains. We can do it on all kinds of organs. Again, reiterating Shauni, if you work on the brain, know that metabolically the brain is very active. You need to be fast. Do not take 2 hours to prep the brain. You need to be within 5 minutes. The brain has to be frozen, which then allows you to section it and so on. So you really have to be fast with in vivo tracing metabolism. I think with this, that was it. It was a bit of a teaser on the tracer imaging mass spec. James wants to have the last word.
I have to have the last word.
Yeah, you have to. Okay.
Okay, so you can stay up here if you like. Do you want to stay up here? No, you want to go. Okay, so thanks a lot again for coming today. That's all of our email addresses there if you want to get in contact with any of us. We're all here all week. Yeah, we're all here all week, so please come and reach out to us. Also, if you want these slides, just come and approach us and we can send them to you. If you spotted the 4 spelling errors that I've spotted, I've already changed them, so that will be fixed for if you want them. So I'll just leave you guys with this last slide before taking questions. We're open to all feedback, including if there's anything that still leaves you scared about doing stable isotope labeling experiments. We're again here to talk and try to allay your fears. Also, do you have the tools you need? Do you have the software that you need to do the work? And hopefully we'll come back again in a future Metabolomics Society conference. So if there's anything that we haven't talked about today that you'd like to hear us talk about in the future, then please let us know. So I'll leave it there, and if there are any questions, then we're here to answer them for, I guess, the next 25 minutes if anybody has anything to ask now. So thanks again for your attention. So any questions?
Hi, thank you for the workshop. Might be a simple question, but I'm not that familiar with tracing. So when Dr. Loopmans presented about like how exactly you should, the possibilities that you could choose to build your experiment, I wasn't completely aware on why the kinetics on the intravenous is the most controlled one. So if you can talk a little bit more on that, I appreciate that. Thank you.
So yeah, when you're giving infusion, You're basically giving the tracer continuously. So the animal or the human, the system will get used to getting this tracer, and all your metabolic pathways will reach a steady state. And that's the most important if you're interested in those long-term kinetics. Like I said, there are people that don't necessarily want to look at long-term kinetics. They are more interested in, okay, what is the first response of the body when I give tracer? And then these bolus injections are better. So it really depends on your own biological question.
Okay, anything else? Yeah, hi. By the way, those microphones aren't very loud, so I can't hear that well, but we'll get there.
I will try my best. Can you hear me now?
I can kind of hear you, yeah.
So thank you so much for the workshops. It was very helpful for me because in my PhD, I'm trying to generate a human reference metabolome. And therefore my question is, do you have any experience with labeling lipids in human cells or mice in general? That would be very useful because I don't have any experience with that, and it takes a very long time to generate this fully labeled reference metabolome. So, yeah.
Yeah, okay, and that was in vitro or in vivo?
In vitro.
In vivo?
No, in vitro.
In vitro, sorry. Yeah, okay, great. And that was just mammalian cells,
Yes.
lipid labeling.
Yes.
So I don't know if anybody else wants to start. So I've specifically talked about fatty acid labeling rather than intact lipid labeling because it is more complicated. And I think one of the most challenging aspects is because you'll be using LCMS as well, you're going to get a lot of co-migration or very close to co-migration. You're also going to be getting label incorporation into the glycerol backbone. So when you get that labeling, that becomes a +3 usually. You may be getting some label incorporation into any head group. So for example, with phosphatidylinositol, you can easily get a +6 from the 6 carbons of the inositol. So all of a sudden, we're not looking at fatty acid chains yet, and yet we're already seeing a pattern of +3 or +6 or +3 and +6 to make 9. And then you've got the labeling of the fatty acids on top of that. So things can get very complex very quickly. And I don't think there's that much of an easy solution other than hard work and exploring that data very closely. I think this is such a basic comment that I'm going to make, but it's something that I've seen people do too many times, so I will say it anyway. It's imperative when you're doing lipid labeling studies or label incorporation into lipids studies that you've got to have an unlabeled control.
Yes.
Absolutely, because that's the only way you're going to be able to tell whether your +2 labeling isn't actually just simply two more hydrogens in an unlabeled fatty acid. Does that make sense?
Yes.
Because things get very complicated with the +2 patterns of label incorporation and -2 patterns of double bond number. So it is complex, it is doable. The only thing I can really add to that is trying to get the best chromatography you can on your LCMS study, and that unlabeled control is absolutely essential. I don't know, anybody else have anything else to?
Maybe something that I want to add is also the time frame that you're labeling. Because, for example, if you look at glycolysis or the TCA cycle, people usually label for 24 hours. For lipid labeling, start with 72 hours because those processes take much more time. So especially if you're interested in having as much labeling as possible, try to go for longer times.
Okay.
Yeah, so when I was talking about my Toxoplasma gondii work, we had 24 hours. I wouldn't, if I had the choice, I would be labeling for much longer than that to try and get more label incorporation. Totally different to the polar stuff, which, as you saw, could be maximally labeled in just a few minutes, just a few seconds in some species.
Okay, thank you so much.
Thank you. We've got two more.
Quick comment and question. So, you're talking about M+2 versus 2 hydrogens. If your instrument has high enough resolution, that can be resolved, right? So it comes down to data tools.
Yeah, absolutely. Absolutely. That's the dream. But unfortunately, I've done too many experiments where I couldn't resolve it that well. But now we've got a proper lipid expert coming to talk now.
I don't know about that. Sorry. Yeah, I think I agree with you that lipid incorporation is really, really tough, but you can do it. I have a more basic question. Are these tools available online yet, especially the Manic? Because I couldn't find it terribly easily.
Especially, what was the last bit? Are they available
the Manic online? Can we get access to these tools yet?
Manic?
Yeah.
Or Maniac?
Maniac, sorry, Maniac.
It's not available online, but dropping a line and we'll share.
Great.
Yeah.
Wonderful. Thank you.
Yeah, but in terms of the talks, yeah, we're happy to share those slides, and there are a few QR codes dotted about as well for access to some of the other software that we talked about. Any more? Yeah.
So I have one technical question about Maniac. So I can use any software for peak picking, such as MZMine, for instance, and then use the quantitation table as input for Maniac. Would it work? Have you tried that?
No, we've just, because we standardized it as our internal workflow, so we've just stuck with Leuven.
So even the peak picking would have to be done using your workflow. Okay, thank you.
But Leuven is an open source software. Yeah.
Okay.
The whole idea is that it's open source, and so
Okay, thank you. And if I may have one more question, as you are asking whether we have all available tools, so all the examples you are showing were about primary metabolism. I would like to ask about isotope labeling in specialized metabolism, where, for instance, you want to see you are in an untargeted mode, so you may be detecting hundreds of thousands of features produced by some bacteria, and you would like to see those metabolites, those specialized metabolites, into which, for instance, a specific amino acid incorporated. So if there is a tool that might be used for this.
Dave? So the question was about what software would you use when you're doing untargeted studies where you're just looking for those metabolites or molecules that have got label incorporated into them?
So, I don't know. I think there's 13CXCMS that's out there. I don't know. I haven't.
So you probably would have to do some. You would have a labeled and unlabeled experiment and do some comparative metabolomics. But I'm not aware of anything that could how to do that other than manually.
It's not something that we tend to do a lot, mainly because it is, you're looking for very defined stuff. So, I mean, I can see where you're coming from. It is something that we've been looking at doing. I don't think we've probably done it more in the GCMS space than the LCMS. I don't know. The last thing I saw was something like. Has people seen 13CXCMS? Or
Yeah, maybe. Maybe
that was around. But yes, exactly. You'll need an unlabeled and labeled to be able to distinguish the shift from what the unlabeled is.
Okay.
Yeah.
Thank you.
Yeah, there are definitely some tools around for GCMS work as well. Okay. Any more? Yeah, one more.
I'm going to do a seamless plugin here. So we actually have two papers published on software called KHIPU, K-H-I-P-U. So that organizes the isotope labeling pattern in untargeted, regardless of what pathway or what experimental design you have.
Okay, great. Well, probably come and talk to you about that afterwards. It sounds good. All right. Any more? Okay. Well, we might be nearing the end. So thanks again for coming to this session and also coming to a session that starts at 8:15 on a Sunday. I greatly appreciate that. I hope you all enjoy the rest of the conference. And please remember, all four of us are here all week if you have any other questions. Thanks so much. Cheers.